Okay, so let's just jump right in. Inside every single one of your cells, there's a 3 billion letter code that makes you, well, you. But for decades, the vast majority of this code was a total mystery. Scientists even called it junk. Today, we're going to pull back the curtain on that so-called junk and reveal a hidden world of tiny regulators that are secretly running the whole show.
Now, you probably remember this from biology class, right? The central dogma. DNA has the blueprint, it gets copied into messenger RNA, and that RNA gets turned into proteins, all the little machines doing the work in our bodies. It's simple, it's elegant, and for a very long time we thought that was pretty much the entire story. But here's where it gets wild. It turns out that a staggering 98% of our DNA does not code for proteins. 98%! For years, this was just one of the biggest puzzles in genetics. I mean, why would evolution bother keeping around all this extra material if it didn't seem to do anything? Which leads us to the big question for today. Is almost all of our genetic code just evolutionary clutter? You know, like a biological attic filled with useless stuff? Well, as scientists started to dig deeper, they realized the answer was a huge, resounding no.
The truth is actually way more complex and, honestly, way more interesting. We're going beyond that simple central dogma to uncover the genome's hidden players.
So here's the key. That 98% isn't junk at all. It's being read and copied into this incredible variety of molecules called non-coding RNAs. These RNAs don't build proteins, but they do something maybe even more important. They control the genes that do. They're the managers, the directors, and the regulators of the entire genome.
And these non-coding RNAs really come in two main flavors. To make it simple, let's think of one group as really precise, single-purpose scalpels, and the other as versatile, multi-tool Swiss Army knives.
So on one side, you've got the small non-coding RNAs, with the most famous being microRNAs or mRNAs. These are the scalpels. They're ancient. We find the same ones in everything from worms to humans, and they follow this really clear, well-understood path to do one specific job with amazing precision. Then on the other side, you've got the long non-coding RNAs, or lncRNAs. These are the Swiss Army knives. They're much newer on the evolutionary scene, they're incredibly diverse, and their mechanisms are super complex and still a huge frontier for research.
What's so cool about this timeline is how it shows the history of the field. See the blue line? That's the mRNA story. The first one was found way back in '93, and that just kicked off this cascade of research defining how they work. The orange line, for lncRNAs, actually starts around the same time, but progress was way slower. It wasn't until these massive projects like ENCODE kicked off in 2007, this huge international effort to map out the entire human genome, that the field just exploded. And honestly, we're still just scratching the surface.
And this visual just illustrates their different evolutionary paths perfectly. Over on the left, you can see how one specific RNA family is found across this huge range of animals. Humans, flies, worms. They are ancient, reliable tools that nature keeps using. But now, look at the lincRNAs on the right. Their numbers just explode as you move up the evolutionary tree. They're constantly being gained and lost. They are brand new inventions, often specific to just one species.
So how does the cell actually make these different regulators? Well, this is where that analogy of a production line versus a custom shop really comes to life. Okay, this here is the miRNA production line. It's this really beautiful standardized process. You start with a long primary transcript, it gets chopped by this molecular scissor called Drosha inside the nucleus, then it's shipped out to the cytoplasm and chopped again by another enzyme called Dicer. And what you're left with is this tiny 22 nucleotide long molecule, ready to go to work. It's like a factory, it's incredibly standardized and efficient.
Now let's head over to the lncRNA custom shop. And yeah, it's a completely different ballgame. There is no single pathway. Some of them are made just like protein coding messages, with a cap on one end and a tail on the other. But others form these complex structures, or even loop around to form perfect circles. All this variety in how they're made leads to this huge diversity in their shape, and, as we're about to see, their function.
All right, so we know what they are, we know how they're made. Now for the main event. What do these molecules actually do? How do they run the cell?
So let's start with our scalpel. A microRNA has one primary job, gene silencing. You can see here how its seed region, just a few letters, finds a perfect matching sequence on a target messenger RNA. And when it binds, it basically tells the cell's machinery, hey, either chop this message up or stop it from being read. It's a simple, elegant little off switch, and thousands of these switches are constantly fine-tuning our biology.
So yeah, if miRNAs are the scalpels, then lncRNAs are absolutely the Swiss Army knives of the cell. They are equipped with a whole range of different tools for completely different jobs, and they often work on a much, much grander scale.
Okay, get a load of this. This is the lncRNA called XIST, and its job is nothing short of architectural. In female mammals who have 2 X chromosomes, XIST literally paints one of those entire chromosomes. It recruits proteins that scrunch it all up and shut it down completely. This whole process prevents a lethal double dose of genes. So you've got one single lncRNA silencing over 1,000 genes all at once. It's incredible.
But lncRNAs don't just act as switches. Sometimes they're the builders. This is NEAT1, another famous one. It acts as a structural scaffold. It just gathers specific proteins all around it to build this little organelle inside the nucleus called a paraspeckle. These structures are like organizational hubs that help the cell deal with stress. So in this case, the RNA isn't the message, it's literally the foundation.
Now here's a third, totally different function, the decoy. This lncRNA, NORAD, is just covered in binding sites for a specific type of protein. By acting like a molecular sponge, NORAD soaks up all these proteins, which stops them from going around and causing trouble elsewhere in the cell. It's this brilliant protective mechanism just to keep the whole genome stable.
So we've gone from thinking of all this DNA as junk to realizing it's a bustling universe of complex regulators. This isn't just a biology lesson. It's a fundamental shift in how we understand life itself.
So let's do a quick recap of the 2 worlds we just explored. You've got mRNAs, the small, ancient scalpels. They're precise, they're conserved, and we understand them pretty well. And then you have lncRNAs, the large, fast-evolving Swiss Army knives. Versatile, complex, and a thrilling frontier of biological mystery.
And why does any of this matter? Because this hidden layer of regulation is at the very heart of everything from how an embryo develops to complex diseases like cancer and neurological disorders. By understanding these non-coding RNAs, we are unlocking completely new ways to diagnose and maybe even treat human diseases.
We've gone from thinking 98% of our genome was just useless junk to realizing it's a command center running the show. It really makes you wonder, as we keep exploring this genomic dark matter, what other incredible secrets are still just waiting to be discovered.